Second chapter, important issues are there for FA1 revision. Second chapter, the cell basic unit of life. This will be asked in FA1 revision. And let us see the syllabus, what it is included. We will have a look on that. About the story of the microscope and history of microorganisms, you need to focus. And this was discovered by Robert Hooke, and along with that Athanasius Kircher and James Swammerdam, Antony van Leeuwenhoek, who discovered this all microscopes and the basic of microscopes. You need to have a look on that. Usually, and this was discovered by Robert Hooke. This is the oil lamp Robert Hooke's microscope. Earlier to this, glass normal single glass microscope discovered by Antony van Leeuwenhoek, but this one it is Robert Hooke microscope, which has oil lamp and again basic lenses here and a barrel which we can see microorganisms. Then this was the Robert Hooke's first observance of the oak tree bark. Then he has seen same like honeycomb structures. Then he founded that cells might be in this form only. Then these cells are called as rooms here. He first used the term called as rooms. So this was the first bark image of oak plant, oak tree. You can see here this was a structure discovered by Robert Hooke from an experiment. And then next you need to see the onion peel activities. To see the cells in the onion, we need to cut off the peel, and after cutting the peel also, here we need to take the fraction of the peel, and with the help of our hands we need to remove that, and then the peel will be there. That if we go on focusing under microscope, then we can see this kind of picture. So you can see cells here, many cells are there. In the cells also middle you can see the nucleus. Here nucleus is there, here nucleus is there, here nucleus is there. These are all arranged horizontally like bricks in a wall. So later, after discovery of the microscope by Robert Hooke, then these all things slowly it was known. Requirements also you need to remember while searching for any activity. You can see here, while taking this onion plant, then onion bulb of onion, we have taken a fraction of that, and then we taken into the hands. After that, we just peeled out the layer. Then we focused under microscope. Just we need to put this over a slide with a cover slip. After adding glycerin, then we can see the cells like that. After finding the cells, you need to draw in your book like this. Understood? This is the diagram of the cells in the onion. Then after that, we have cheek cells also. Cheek cells we need to scrap with the help of scraper inside our mouth region. Then we need to put our microscope. That means with the help of cover slip, slide, then glycerin. After that, we will be observing some bigger cells and a nucleus. Can you see here, the nucleus is very bigger, and this is a cell membrane, and this is jelly like substance inside that, it is known as the cytoplasm, and this one is known as nucleus. Bigger nucleus is there. After observing that, you need to draw like this. So these are the cheek cells which are present in our body, in our mouth, and this you need to remember. After that, we will be having about Robert Brown story. Robert Brown, name ends with N. Robert Brown, N means nucleus here. That means to remember, we are discussing. Robert Brown discovered the nucleus, and the structure of the nucleus was differed in different kinds of plants. And these are the plant cells which we can see. Stomata also. This is the internal structure of a plant cell, or the leaf structure we have seen. You can see the nucleus. Here is the nucleus, and this is the cell wall which is present in a plant cell, and these are the stomata region. You need to have a memory on that, inside that. And here you can see different kinds of the chlamydomonas, amoeba, and here bacteria is there, and the seagrass, chloroplast, and here also we have cells in a plant cell. Chlamydomonas is a single celled organism which can live in the water, and amoeba, it is a single celled organism. These both are unicellular organism, and this is bacterium. We cannot say cell. Why? Because nucleus is not perfect in this organism, so that only they cause many diseases, it seems. And then next here, plant cells, these are all multicellular organism. That means basic organism, unicellular organisms are there, and multicellular organisms are there. Unicellular means here chlamydomonas, and here amoeba. Amoeba consists of pseudopodia. With the help of pseudopodia, then it goes on eating the food, and it has contractile vacuole. This one is known as contractile vacuole, and it is a exhausted food material, and it has nucleus here. One nucleus you can see, and these are total irregular pattern shapeless microorganism. You can draw it in any shape, but basic parts you must not miss. And this is the chlamydomonas, and this one is known as uh, plant cell. This here in the, here in the indicating about xylem and phloem. Then you have one more cells here: spindle cell called as muscle cell, strip to muscle cell, and bone cell, and the nerve cell. So these you need to prepare for your exam. Most important topics. Apart from this, the basic questions cannot be coming. I am making it guarantee in your S7 summative assessment 1 also. Complete these questions only will be based. Understood? And then next here you can see blood cells also, RBCs and WBCs, and then these are called as erythrocytes, these are called as granulocytes, and these are called as agranulocytes. Granulocytes and agranulocytes means here in granulocytes, can you see here granules are present, but here not that much bigger granules are present, plain only it is there. Small are there, but they are negligible. So these are basophil, neutrophil, and eosinophil, lymphocyte and monocyte. These cells in ninth class you will be getting very descriptive classes on this. Understood? So you need to focus on this cells which are present in our muscles. You will eat chicken, so that is this muscle, chicken muscle, and bone also you will eat sometimes, that is bone cell. And chicken and mutton if you are eating means that this is the thing you are eating. So these are all present in our body only. In our body, and humans and animals are present, and these you need to remember the diagrams. And the plant cells you need to remember, and Robert Brown you need to remember, and this cheek cells diagram from the experiment, and onion cells diagram from the experiment. And then next you have onion peel activity. Then next you have cork cells by this microscope which was invented by Robert Hooke. Bark, onion peel cutting, onion peel layering out, onion peel focusing under microscope, onion peel cells, cheek cell diagram. And next you have Robert Brown, nucleus, plant cells, stomata, and chlamydomonas, amoeba, bacterium, plant cells, seagrass, grass it is grass, muscle cell, strip to muscle cell, bone cell, nerve cell, and these are the blood cells. And last you have about the comparison between the units. So it doesn't mean that this is the bacterial cell, this is the human cell, this is the elephant. So maybe the number of cells are more in elephant, but the function and the structure and similarity will be same, only number will be differed. Understood? All the organisms will be made up of cell only, but number will be differed. Here you have some data. 1 meter equal to 100 centimeters, unit is cm. 1 centimeter equal to 10 millimeters, 1 millimeter equal to 1000 micrometers, or called as microns. 1 micrometer equal to 1000 nanometers. These topics you need to, nanometer unit is nm. And these topics you need to revise for second chapter. Apart from this, nothing will be asked in your exams. And third chapter also will be there, and third chapter revision we will continue.